Hello, everyone. My name is Robert Liu. I'm a scientific director from GSK in the BioPharm process development group located in Pennsylvania, the United States. Today, I'm going to talk about the small-scale model justification, an industry position paper. I'll be supported by my team members, and they will carry out the question and answer session after my presentation. And this presentation is about a white paper. This white paper was published in May last year by the Qualification of Small-Scale Model workstream in BioPhorum. Our workstream team has about 26 companies, over 100 participants from 14 countries. We have published a paper last year, and after that, we have been presenting the overview of the paper at different conferences. In this presentation, I will talk about the qualification of a small-scale model and the process characterization workstream. The small-scale model workstream has been concluded, and we moved on to a workstream with process characterization. Then we're going to talk about the current regulatory guidances on small-scale model qualification. Then about the paper itself, the justification of a small-scale model in industry perspective. And you can see the white part has the contents of the paper, then the conclusions. Acknowledgement as well. So the first about the workstream, and as we mentioned, the small-scale model workstream has been working, and we have published the paper and presented at different conferences. So now we're moving towards the process characterization. So this group, this workstream is forming, and we're drafting our chapters for this process characterization workstream. And there are other workstreams in the BioPhorum development group listed here. Next, I'm going to talk about the current regulatory guidances on small-scale model qualification. And in the guidances, we know the small-scale model are utilized to meet the regulatory expectations, and we know for the manufacturing processes from ICH, and also established control strategy covering continuous process verification and life cycle management, also from ICH, and also support manufacturing process development and validation. There are high-level regulatory guidances exist from other agencies, for example, FDA, on process validation general principles and practices from 2011, and also EMA guidances on process validation. However, there are no detailed guidances on how people carry out small-scale model qualification work, and our group thinks this is a gap. Therefore, we wrote this paper and hope the paper will provide an overview of current practices of the qualification of small-scale model used for development of biopharmaceutical drug substance manufacturing processes. So next, I will talk about the paper itself, justification of small-scale models and industry perspective. So here we have a link to the BioPharm website. When you click the link, you can find the paper and you can download it as well. And so the paper, since we have the white paper, is a long paper, and we also break down the white paper into 3 parts as listed here, and we publish on online bioprocessing journal. And in addition, we have gone to different conferences to present, for example, American Chemical Society, and also Bioprocess International East. And so this seminar is also one of the presentations we give into the BioPharm in the global community. So now I'm going to talk about the purpose and scope of the white paper. So the demonstration of the small-scale model is representative of a large-scale manufacturing system is important. And this can support process validation, and it's required by the regulatory authorities. So it is important to understand the degree of which those models represent the commercial process. And we aim to provide options and tools for the small-scale model qualification in design, execution, data analysis, and justification of results. We also provided case studies for upstream and downstream unit operations. And the topics on process characterization are not in scope because we have another, as I mentioned, the workstream on process characterization following up. So here are some terminologies of qualification and assessment. So the small-scale model, any scale smaller than the full-scale commercial process. And the EMA here requires a small-scale model must be designed and executed and ultimately justified as an appropriate representation of the manufacturing process. Here also mentioned is a small-scale model qualification is a formal procedure to justify representative behavior of a small-scale model in comparison to manufacturing scale. The requirements for each small-scale model qualification are based on multiple aspects and may differ between companies and between projects. For small-scale model assessment, it is an effort to justify representative behavior of a small-scale model in comparison to manufacturing scale. Small-scale model qualification is a more rigorous form of the small-scale model justification. This figure here shows the application of small-scale models, and it can be used across production development. You can call it a product or process development lifecycle, and it's representative of the large-scale process, and the justification increases with the maturity of the product development. The qualification of a small-scale model are heavily used in stage 1 and stage 2 of the process validation lifecycle. For example, in stage 1, we have process characterization studies. Then it can also be used to support post-approval changes and the deviation investigation in the continuous process verification stage. There are special considerations for viral clearance support, and this table shows different considerations of different steps. For example, here the unit operation with chromatography, viral filtration, and virus inactivation, and listed the different parameters, scale dependent, independent, operation conditions, and what stage we have. For example, in the US, we have R&D filing for clinical phases, and also we have BLA filing in commercial examples. So this slide shows the qualification is related to quality oversight. So for the small-scale qualification, normally there is a little to no quality oversight in the early phase of the product life cycle. In later phase, some company would use quality oversight and some company would not. So it's a case-case determination. And our survey shows the QA oversight may enhance overall small-scale model acceptance. So now I'm going to talk about the design considerations. First is a small-scale system versus large-scale system. And we would like to carry out a risk-based design for small-scale model justification. And I'm going to talk about assessment, qualification of small-scale model, the number of runs needed, acceptance criteria, evaluation criteria, and the use of statistical methods. So those all the contents are covered in the white paper. And here we only give overview of the white paper, and you could get more details in the white paper. So this shows the small scale system. We usually for process development, when we scale up to a large scale system, then we scale down for the small scale model qualification work. And this shows more details of for upstream process scale down approach. And there are two approaches there. One is called a satellite approach or paired approach. Another one is called non-satellite approach or unpaired approach. So there are pros and cons for each approaches. The small scale model batches is inoculated N-1 or N bioreactor at scale. So this is for the paired approach. And these scale dependent differences from the random variability can be isolated. And this one is good to detect the potential differences between the scales. However, this approach requires good planning because we need to arrange our small scale align with the large scale timing. For the second approach, the non-paired approach, this one does not require inoculum transfer from large scale to small scale, and the small scale seed trains are passively qualified and is uncoupled from large scale runs, and execution is less complex. So because we don't have to align the timing from large scale to small scale, however, the capability to detect potential differences is lower compared to the paired approach. This slide shows the downstream process scale down approach. It's a paired approach. So what we would like to do is to take the load material from the previous steps of the manufacturing process, and it is utilized for small scale model qualification. And we take, for example, from the large scale run 1, we take the material, then we run small scale. And similarly, from large scale 2 and 3, we also take the material that run the small scale model qualification. And then we compare those processes. And so this allows us to use the similar starting material for the large scale and small scale. Now, once we finish the lab work of the small scale model qualification, and we need to have samples and send for analytical testing. So we have some considerations here. They are IC dependent. Some people use the QC, some people use non-GMP sample analysis in the development group. And intermediate precision should be controlled. And the operator with the training will include the different type of considerations, because if we use the different laboratories of testing, and if the small scale and large scale samples are measured at different laboratories, comparability assessment should be performed before the different analytical labs. And so the guidelines should be applied for different labs as well. So now, after we qualify the small scale model and moving forward during the life cycle of the product, we may need to requalify the model if we have the changes in manufacturing site, manufacturing scale, and also if there is a quality attribute assay changed. If we have those changes, if we do not qualify or requalify the model, and then we should justify why we do not requalify the model, and that justification should be documented. So next, I'm going to talk about the data analysis and the statistical methods. Performing small-scale model qualification, most companies we surveyed employ a statistical evaluation of the data to compare small-scale model with the large-scale system. And the outcome of some of the small-scale models are binary, so they would give you is comparable or not comparable from the statistical analysis alone. However, those outcomes must be evaluated by SMEs in the totality of the evidence when we decide if it is qualified. So those methods, if the statistics are used, should be adapted for both satellite approach and the non-satellite approach. So there are different types of statistical analysis, and there are advantages of disadvantage for each method. The type of method we have is descriptive, inferential, or combined descriptive and inferential method. So for the descriptive method, which most people are very familiar with, this one, we only take samples and there's no inference to the unknown population at scale or small scale. The examples include the scatter plot, tables listing the numbers of data points, etc. The inferential methods is refer to the population. And for example, there's a different tests. And this procedure tests the non-hypothesis. The hypothesis is the mean of the small-scale model, and against the hypothesis of the alternative hypothesis is that the mean of the small-scale model is not equal to the mean of the large-scale model. And there's a p-value computed and used to decide whether the data is consistent with the non-hypothesis. An example is the t-test to compare the means between scales, and also f-test to compare the variance between scales. And there are some drawbacks of this method, and we're going to discuss a little later. And there is another inferential method used often. It's called the equivalency test, or two one-sided t-test or toast. And so this test is tested the difference between means of the scale and small scale, and also if they have practical differences or not. And theta here is called equivalence margin, and we test the means of the small scale and means of large scale, the differences between them, and to see if they're within this margin. So there are some differences of the t-test and also the toast. The disadvantage of the t-test is the t-test at the beginning assumes what is intended to establish, which is the equivalence of the small scale and the large scale model. They assume is already established. It does not consider what the practical meaningful differences is, and also it incentivizes the small number of samples. So those are the drawbacks of the t-test. So there are advantages of the using toast. So first, the equivalence is not assumed state of nature. This is for the toast as compared to the t-test. So this one is the alternative hypothesis. Secondly, the equivalence margin is determined prior to the data collection, as we mentioned the theta earlier. So this one will bolster the integrity of the procedure. And third, the means are truly equivalent. The chances of the toast rejecting a null hypothesis increases with the sample size. So this one encourages people to use more sample size and get more accurate results. So here I'm going to talk about another approach for statistical analysis. It's called quality range or QR method. So this method determines a quality range using the large scale manufacturing data. For example, we can do a margin of plus or minus 3 standard deviation of at scale data. Then we plot the data from small scale if the small scale is within that range, and we consider this small scale model is qualified. Another method is called multivariate analysis, or MVA method. This method has been more popular recently. And in this one, the model includes the method principal component analysis, PCA, or partial least square analysis. For example, here we have the large scale data set in blue and the small scale data set in red, and we compare them in this region. And for example, here, this one, we consider them comparable. And here, you can see the small scale data is away from the large scale data. We consider them not comparable. So this advantages of using this one is consider multiple time course variables simultaneously across batches. The disadvantage is it requires combination of diverse process data, and to get them together, for example, the growth titer and other product quality contribution. So now I would like to conclude our presentation. A small-scale model is a lab or pilot scale system, a group of equipment that represents a manufacturing scale system. So all the small-scale model need to be qualified, it needs to be justified. Some needs to be qualified using a risk-based approach. Data from large scale are usually used to set criteria or references. To qualify the small-scale model is to demonstrate we have a comparability of the small-scale model population compared to the large-scale population. In terms of output, we can have CQAs, step yield, etc., and using the sample runs taken from both scales. So we should consider the devil is in the details, and to consider which approach to take case by case when we design the study. So in all the cases, we should have sound scientific rationale for the approach and to document them. So the sponsor, like us, pharmaceutical companies, is confident to defend the study when questioned by the regulatory agencies. So lastly, I would like to acknowledge our team, and this slide shows the lead authors of the paper and also additional contributors of the white paper. Those are all from the small scale model workstream team. And also there are other colleagues from different companies also on the team. So, then there are the antitrust compliance statement from BioPharm, and also supply interaction policies and code of conduct from the BioPhorum. So this is the end of my presentation. Thank you very much for your attention. Thank you.